So today, our speaker is Lina Fajardo from the University of South Florida. She's talking about digraph homology and DNA rearrangement. Over to you.
Well, thank you for the invitation. So to give you an overview of what we're going to do today, and I'll move faster and slower depending on how we're doing on time. Goal is to give you motivation for everything because it will be mostly abstract mathematics. It will be completely or almost completely abstract nonsense, but all of this nonsense is rooted in something that we're trying to study. And so the discussion in the end and where we're trying to go with all of this will come back to the biology in the end. Once I've grounded that, I can tell you about what these word graphs are, how we study their homology with a non-simplicial kind of homology, Prodsimplicial homology, what we've been able to find in a way of generators, and some final thoughts on where this is all going. So our model, so this is all meant to study DNA rearrangement, and specifically we're studying ciliates. The special thing about the ciliates we're studying, in our case, Oxytricha trifalax is the model organism. Most things that mutate their DNA a lot get cancer. Oxytricha just thrives with DNA rearrangement. And what we've been able, we as a lot of people, what biologists have been able to figure out is that it has two kinds of nuclei. So it has what we call micronuclei and macronuclei. These ones are not transcriptionally active, so all the information to create proteins and so on is not actually available here because the information is cut up into pieces, scrambled, jumbled out of place, sometimes inverted. But what we know is that during reproduction, it deletes this and it deletes this and is somehow able to recreate it from the jumbled up mess. And what we're trying to study is how it goes from this precursor strand where everything is mixed up to this nice, useful product strand that it can read and create proteins from. The model for this mathematically is we label the segments, we number them, and biologically we know that every gluing point, so these points here, have repeats of DNA. So in the DNA sequence, we see repeats here and here, where these are going to glue. And so there's only one retained here. Those get names. Those are pointers. They're colored here for my own amusement mostly. And what we want to do is model how this DNA strand bends itself so that it can have all of these segments adjacent to each other and in the correct order. And what would happen is at each of these gluing points, it will cut the junk DNA that doesn't have anything that's useful. One way, so the precursor to everything that we're doing here, is to model this with assembly graph. So you pretend that the DNA strand, the long one, can contort around itself, and at every one of these gluing points, which we have labeled as by order of appearance and the product strand, you can delete certain patterns that appear. So depending on how I delete the patterns, what I'm doing is I'm doing smoothings of these vertices. So these are called assembly graphs. These are not my work, but this is what my work is based on. And what I want to do is find ways to model this smoothing behavior, the smoothing mechanism, where the DNA joins and then cuts out the non-useful strand, so that in the end, we can have this nice, clean sequence where all of the pieces are concatenated in the correct order in the right direction. So what we do is we go back to the pointer sequences. These are labeled. And because I have two gluing points for each of these results in the product strand, I'm going to have exactly two instances of each of these labels. What that creates is what we call a double occurrence word. So one word where every symbol appears exactly twice. That's the definition we see here. Similarly, we can also define single occurrence words, and we can write down the alphabet or sigma, and then the words that have at most n symbols that are double occurrence words with this symbol, if they're single occurrence words, then with this symbol. It appears maybe a couple of times in the proofs, but because it will be at points notation heavy. This is just good to have for reference. If you need me to, I can always come back to these slides. Now, what we're going to do is, if you look at all possible double occurrence words on any kind of finite alphabet, it very quickly gets out of control. So what we're going to do is we're going to consider them up to ascending order, and then we're going to create equivalence classes. Ascending order is where I write the words in such a way that the first symbol that appears is in the ordered alphabet, the first symbol available. So I don't go to the symbol number 3 if there's an earlier symbol in my order, like 1 available. Then if I need a new symbol next, I'm not going to go to 1. The next symbol will be exactly 1 more than the last highest symbol I used. And I can say that 2 double occurrence words are then equivalent if I can write them down in the same way when I use ascending order. The patterns that we're trying to delete in the assembly graphs correspond to repeat and return words and double occurrence words. I'm going to use superscript R to denote the reverse of U, so the word U, where all of the symbols are in the opposite order. I can write UU as a repeat word if there are 2 instances of U embedded somewhere in the word W, so it's a repeat word in W. Otherwise, I can also have U reverse be a return word if both U and U reverse are embedded somewhere in W, possibly interrupted by something, but these words could also be empty. The important result and why we're interested in these is that iterated deletions of these explain a lot of the rearrangement that we see in Noxotrica. So again, this has to be grounded in something. The motivation is to explain these DNA rearrangements specifically in this organism, but towards the end, we can change what the rules are for reduction. Specifically, we're not going to be reducing just any double occurrence word. We usually go for maximal double occurrence words. So maximal repeat or return words, and I have notation for what those are. In the end, what I'm going to be interested in is finding ways to reduce long double occurrence words. So it would be the equivalent of my long full assembly graph. And as I smooth my vertices, I get reduced double occurrence words that still represent the same DNA molecule. So what I have here is I can do immediate successors of a double occurrence word if after deleting a maximal repeat or return factor, I get this new word, du of W. For example, let's look at this word. I can see that my double occurrence words, the maximal ones, are 1, 1. So this one is a trivial one. It's either a repeat or return word. It won't matter because it's length 1. I have the repeat word 2, 3, 2, 3, and I have the return word 4, 5, 5, 4. If I look at only the single occurrence words that appear in repeat or return patterns, then that would be these ones. Each of these I can delete in its own way. When I do that with 1, 1, when I do that with 2, 2, what would be remaining is the letters here in black. If I do that with 4, 5, 5, 4, and then what I want to do is call those the immediate successors of W. I delete the 1 first, and the remaining word I rewrite in ascending order. So I'm not going to hold on to this longer than necessary collection of words. I will only use them up to equivalence. Same thing goes for the next one. And now I still have to slide. So the tab is not working too. And then the last one, if I delete 4, 5, 5, 4, gives me this other word. With these deletions, what I'm going to build is a word graph. So I'm going to create a directed graph that holds on to all of the double occurrence words as vertices and gives me an edge between them if I can find a deletion of a maximal repeat or return word that results in the new one. So with the same word we were examining a moment ago, every time I delete something, I get 1 new edge. And then all I do is the first 3 successors of this initial word are the ones that we did in the example. And if I repeat that process a few more times, I should eventually end up with the empty word. Here you see the reduction, then you see the rewriting, reduction, rewriting, reduction, rewriting. If I consider all of the possible double occurrence words of size up to n, I can build a global word graph. So I can have this larger than life kind of graph, where everything is included. And then what I'm going to look at are the rooted word graphs. So from one particular word, double occurrence word that I'm interested in, I can go down to the empty word. It has a partial order. You can do other kinds of combinatorial things that way, but sometimes it might be of interest to study the entire word graph. So here, for example, we have just smallest example because they grow very quickly. The word graph on 2 symbols. Here are all the possible deletions, all the possible layers available. And this notation is what I'm going to use for my directed edges. So sometimes you might see just parentheses, but I want to hold on to the order and the fact that these are going to be simplices later on. If I look at a rooted word graph, so for example, this is a part of the word graph, the global word graph on 3 symbols, but I'm rooting it at 1, 2, 1, 3, 3, 2. And I can see all of the successors and then follow those all the way down to the empty word. It's on these graphs that I want to compute homology groups, but I'm not going to use traditional simplicial homology. I'm going to use Prodsimplicial homology. And the motivation is again related to biology. It relates what would be equivalent pathways for smoothing over the vertices. So if I define the building blocks for my cells, I'm going to use what I'm calling consistently directed graphs. So these are directed graphs with a single source, a single target. So one vertex that has only outgoing edges, one vertex that has only incoming edges, and no cycles inside. So something of this sort, where I have a cycle, even if there's a source and a target, would not be allowed. I'll do big cross no. But otherwise, this would be okay. This would not be okay. Yeah. With this as my building block, I can define simplices in such a way that they're number 1, directed graphs. That's easy enough. But number 2, they're also consistently directed. So I can do the same usual definition by induction. You do just one vertex, and then every time you go up 1 dimension, you add 1 more vertex, and you make sure that the edges are pointing directly towards the new vertex. So I don't just assign these directions willy-nilly. I always make sure that the new edges are pointing towards the new vertex. More than just simplices, though. The selling point here is we want to use Cartesian products of simplices. So that's the Prodsimplicial and everything. Here I have the Cartesian product of 2 directed graphs. The formal definition is here. But in intuitive terms, what I want to do is create 1 copy of the first graph for every vertex and the second one, or 1 copy of the second graph for every vertex and the first one. So you can see that there's either 3 copies connecting corresponding vertices in the triangle, or there are 2 copies of the triangle connecting the 2 copies of the edges of the vertices in the single edge. This Cartesian product is associative, it's commutative up to isomorphism, and it preserves this consistent direction of graphs. So it preserves what we want out of our cells. They are primes, so if you were trying to break something down so that there was a product of non-trivial graphs, you can't break things down into anything smaller than simplices. That's also going to be computationally convenient. And the factorization is unique up to the order of the factor. So because this is commutative, it doesn't matter which way I order them. But I'm going to have almost everything else I want. With this, I'm going to define the Prodsimplicial cells. So officially, I'm abusing the notation, right? Because this is the Cartesian product, and this would only be the 1 skeleton. I only have the wireframe of all of the edges that are produced by the product of the graphs. But what I'm going to do is assign to this the completely solid cell. So what I'm doing is on this wireframe that is just a graph, that I started out with, every time I run into an instance of something that is isomorphic to a product of directed simplices, I fill it in. So I'm going to say that this is equal to the product, even though it's not really true. To see an example of how I would endow an arbitrary directed graph with this definition and turn it into a proper simplicial cell complex, we have in the beginning just the vertices, then the edges, and then we keep adding more things every time that the previously defined cells allow me to create a new one. So on this one, I start with my graph. This is just a wireframe. First step, I look for only the vertices. I add those in to my complex. Then I also make the edges solid. So far, I still only have the one skeleton, but then on the next step, I'm going to be looking for anything that would be a product of simplices. In this case, the only products available would be one edge times an edge, so that I can get dimension 2. Those would be the squares. So they would look like this or like this, as long as they are one edge going this way and one edge going that way, I'm going to get this consistently directed square. And every time I find one of these, I'm going to fill it in. So that would result in something like this. What you see now is that, for example, this space here between these vertices was not filled in because I actually have 2 edges going this way and then 3 edges going that way, which is impossible to create as a product of simplices. If I go up more dimensions, I would be looking for something like either tetrahedra or cubes or triangular prisms. There are none. So in this case, I stop the building here. And if I wanted to do any kind of homology computations, I do it on this complex. So I should find, for example, that H1 is non-trivial. I have a loop right there, but I don't have, actually, no, I do have also some non-trivial H2. If I'm going to define homology groups, I also need to define boundary operators. So for simplices, it works exactly the same way as it does in the traditional definition. What I need to do is extend that definition so that it will work on the products. So this is a variation of the product rule, the Leibniz product rule, and it has this adjustment with this term corresponding to the sum of all of the dimensions up to that term. So that when I compose it twice, I get 0 every time. And the notation here is that instead of deleting just one vertex at a time and doing the corresponding simplex, I am doing the boundary over one of the terms and would be this product. So I do what used to be my whole product, and then the jth term, I apply the boundary with this definition. So this is the simplex definition for the boundary operator. So we could prove that it behaves the way that you would want it to. I will not bore you with the details. The proof is exactly the same. But if it works and it behaves the way that we want it to, it's still a linear operator. We can define a kernel, we can define the image, and we can define homology groups. Homology groups can always be broken down into some direct sum of what we're going to call the rank or the Betti numbers, beta n, and some extra terms that sometimes do, sometimes don't appear that correspond to torsion. What it's counting in this case is n-dimensional holes. So like above where I had that space that was not filled in, I had a loop that I could poke my hand through. That would be a one-dimensional hole. Examples of what else could result in holes, I can have either cycles. I can have things that are not consistently directed. So this doesn't have single source, single target, or things that are consistently directed but do not correspond to a product. So this couldn't be a product because if it were, I would need these 2 edges to have the same direction. Then what you do is we dream up what kinds of things would result in holes, what would create non-trivial homology. just in a strictly theoretical sense, if I had nothing but graphs, what kinds of things? So for the first homology, the simplest kind of generator would be these loops, kind of like the pentagon in the beginning, except you could make them as long as you want. As long as you have more than 2 and 2 on each side, there's not going to be a good way to fill this out with triangles or squares. So this will create a cycle. The smallest such cycles that appear when I do word graphs of double occurrence words are these two. So if we were looking for minimal cycles, it will be a pentagon, and it appears in at least these 2 different flavors. From here, what we do is we try to generalize what are the kinds of words that result in this kind of shape. So if not a single symbol at a time, what you can do is try to extend it to what kinds of words would reduce in the same way that this one does. Here, I can only have trivial deletions, so I can only have trivial repeat and return words. There's no longer maximal repeat and return words, but we can hope for longer and then create bigger words that have the same homology. So what it looks like is words that have this form or this form, with U1 and U2 having the same length, and all of these being double occurrence words. For the second homology group, we can have, as some of the smallest examples, something like this. So this has non-trivial homology. These graphs don't appear exactly as word graphs, but when they appear with at least one of these square faces broken down into triangles, but those would be homeomorphic. So for an example of that happening, we have this graph. So if you can imagine this as a single square face, instead of being 2 triangles, then this should be homeomorphic to the graph above. It will also have a non-trivial 2 cycle. The other kinds of small generators for H2 are these ones. They look, I don't know if you want to call them pillows. I tend to think that they look like bananas, but I have these like 3 different square faces, all of them one around the other. They're not quite what you would imagine as dihedra in 3-dimensional space, but they work out nicely. So you can do the algebra with these, compute homology groups, and those also appear, but they don't appear by themselves in any of the double occurrence words graphs. In general, when you're trying to figure out what happens if I have this long double occurrence word, I compute its word graph, I turn it into a cell complex, what kinds of properties do these complexes have? One would expect that bare minimum, if I have a subword, so a successor, if not immediate, at least a couple of lines down the road, that the homology of the successor is somehow less than or equal to that of my root word, and that doesn't always follow. So the counterexample for a lot of things are tangled chords. So there are these words where you invert a couple symbols at a time. So you do 1, then you invert to 1, then you invert 3, 2, then you invert 4, 3, all the way until the end. These are not for nothing called tangled. They represent some of the most scrambled DNA that we have been able to find. And here we can see that the increase doesn't quite work the way that you would expect it to. So these tables are all done by brute force with the Python scripts that I wrote. But what you can see is after one single symbol reduction, so for example, if I delete just the 5s from here, I'm going to end up with a word that in ascending order is exactly the same as this word here. But it's not true that if I go up one more, so if I delete just the 6 here, I end up with exactly this in ascending order. This non-trivial homology is not repeated down here. It goes back to being just 1. And actually it stays 1 for a good while. Something I did not have room for in the table, but interesting maybe if you're into that sort of combinatorics and comparing the bounds for possible values. If you look at the number of vertices in these graphs, they grow very quickly, but they don't quite grow on par with these. So they grow in the beginning a little bit faster and then they slow down. So they don't quite keep up with the Betty numbers. For what you can do to combine words, so if successors are not a good indication for how you're going to predict future homology groups, how else can you mix and match words together? I'm not including here some of the more trivial examples, like palindrome words, of course, always have the same kind of homology. They have the same word graph. I can do very simple square words where I just repeat the pattern. Those are not terribly interesting either. I could do very long strings where I just repeat symbol like 1, 1, 2, 2, 3, 3. That's also not terribly interesting. So next up, if I'm trying to combine words in a more meaningful way, what I'm going to need is for my words to be co-prime. So I want them to not share any symbols. So for a moment, I'm not worried about the ascending order just yet, because what I'm trying to do is do all the possible reductions and concatenate them. If all those concatenations are different, then I consider these words to be co-prime. So if I look at these and these, here I didn't write it in ascending order so that I can combine it with these ones and still result in double occurrence words. By inspection, you can check that all of those concatenations would be non-trivial. Not the same, sorry, not non-trivial. So if you concatenate words that are co-prime, you can at least somewhat preserve the structure. So you can have copies that are not necessarily disjointed, depends on the graph you're working with, but you can have copies of the corresponding word graph. So I can do one copy of something that looks like GW, and then one copy of something that looks like GUU. Oh, hold on. I think I forgot to write here and GUU. There we go. From just those two things, you can build a lot of things actually. So once you can concatenate, if you know how to look for things that have non-trivial homology, so for sure I know that all the tangled cores have non-trivial homology, I can concatenate them if I'm careful not to mix and match too much. I can create arbitrarily large homology numbers, Betty numbers. So what would be interesting would be see if we can find other generators that arise in word graphs. So a lot of this has been done by brute force algorithms. So we computed all of the possible word graphs, all of their homology groups for up to 7, maybe 8 symbols. Around 8, the numbers got unmanageable and my laptop could not do it anymore. But we have extensive numbers to play with. It turns out that the Betty numbers for tangled cores are much higher than average, at least if you're averaging over all the possible double occurrence words. They're also some of the most commonly occurring high Betty number words that come out in the biological data. But we would like to see if there are other generators that arise in word graphs. Maybe we can find them by inspection. Ideally, we will be able to find rewriting rules that don't necessarily make things follow from concatenation or from quick embeddings. There might be other things that we're missing. You could also mess around with how you create the word graph. So there's no rule that says that you have to do exactly repeat and return words or maximal repeat and return words. You don't have to use ascending order equivalents to reduce the number of vertices. I actually coded the scripts to take these things as optional arguments. So when I link to the GitHub that has all of this, if you wanted to, you can also play with this. So you can put in symbols that are not integers. You can play around with different orders. You can play around with what kinds of things get deleted or maybe try doing the other way around. So start with the empty words, see what kinds of insertions you can make, and then what the resulting graph would be. If we're going to tackle the higher order, so the longer double occurrence words, if we're going to do still brute force, just to study what happens, like what about all of the double occurrence words that have 10 symbols, 11 symbols, 12 symbols? Computationally, it gets very expensive. So the hope would be that there are nice reduction rules that you can use. So for example, if I knew that in the same way that 1, 1 reduces to the empty word in just one step, but this is homeomorphic to what would happen if I had 1, 1, 2, 2, because this reduces to 1, 1 in ascending order, and then that goes to the empty word again. So there are at least some rewriting rules. This is almost a trivial example, where this is homeomorphic to this, so the homology groups are going to be exactly the same, but then I can knock out a lot of high order double occurrence words by knowing that this particular reduction is going to preserve the homology groups. So ideally, we would be able to find other ways to manipulate things algebraically or as a semi-group before we compute homology groups so that we can find more equivalences between the graphs. If we're looking for minimal generators, the current state of the art is brute force. There should be a better way, especially when it comes to torsion. So we have found some torsion. That's the next. We have found some torsion. It does not appear until double occurrence words of length 10. It at least appears in the tangled chords. So tangled chords, again, are special in this way, but they do not appear sooner. So maybe there might be a much smaller generator out there that we're missing because we're not really computing everything of size 9, for example. But it would be interesting to figure out what exactly causes torsion. And the other one, so this is going back to the applications. How do Betty numbers compare to other measures of scrambling? So the assembly graphs have been used as a way to measure scrambling. So depending on how many vertex smoothings you need to go from the precursor molecule to the product molecule, that's one way to measure scrambling. You can also just count things like the separations between like symbols and double occurrence words. You just add all of those together, one symbol at a time. That's called separation of a double occurrence word, and that's another way of measuring scrambling. So what we would like to do is compare how the Betty numbers with the Prodsimplicial homology compare to these other measures that are known in biology, and then see if that agrees with what evolutionary biologists think of the scrambling in different organisms. All of this is still work in progress. That's, as you saw, still a lot of open questions. But for any of you, if you were interested in puttering around with them, all of the Python script and the Sage scripts are uploaded onto GitHub. The GitHub includes a link to a, oh, did I forget the name? Okay, the name's eluding me, but a nice Python generated website where you can actually type in double occurrence words. It will create the graphs for you. So that might be something for some of you to play with. An earlier version of this works, so it does not quite have all of the things that I'm including in the talk, but precursor to this is up on archive. So if you want to skim through that, that's available. We're currently working on one that is more pure mathematics, and then another one where we actually show the results of applying this to biological data. So we can actually use it to compare through different species of ciliates and say, does the scrambling, so if I use the sequence of Betty numbers going up in dimension as a topological signature for how scrambled the genome is, can I somehow compare how different two species are? So the hope would be that I can find the species that are found known to be in evolutionary biology closer together. They might have more similar scrambling. If I just ran the numbers in all of the genomes, the context in the genome, I might find on average or as a median number, Betty ones, Betty twos that are closer together. All of this is work done. Since I started my PhD, back at the time, these were the group members. The team has since changed, but this is the most recent picture I have of everyone. So I should also thank the NSF grants that funded my work and my PhD program, linked or actually just listed below. And with that, I'm going to thank you for your time. Yeah, timing's more or less okay. I have 3 minutes to spare.
Great, thank you. Firstly, let's thank Lina. If everyone wants to unmute and clap or express your appreciation however you choose, then let's do that. So thank you. And then secondly, does anyone have any questions? I will start if no one else has a question. So you define these on essentially post sets, right? They're directed acyclic graphs, and you have a source and a sink. So it's kind of a post set with a top and a bottom. But there's no reason that it has to be a post set, right? The condition here is that you're looking for these subgraphs that are products of cliques, but you could do that on any directed graph. It's
Correct.
Yeah. Okay. Cool. And if you do it on any directed graph, then the complex you get will be a subcomplex of the directed flag complex. Is that right? Where the directed flag complex is the complex where every directed clique gives you a simplex.
That sounds correct.
Okay. Yeah. Cool.
Yes.
So this will be a subclass of the directed flag complex. I was just okay. I was just trying to get that clear in my head.
I'm just parsing because I know that at different points we've called these transitive tournaments. They're also directed cliques. They're also
there's a few different terms kicking around for them. Yeah, cool. Thanks. I have more questions. Dave, do you want to show a question?
Yes. The examples you did or computed were relatively small. How large is the genome you're working with? If you have one of these organisms, how large would the graph be? How many nodes?
They get exceedingly large. So some of the things we have just not been able to compute. For example, some of the contexts have in the double occurrence word upwards of 130, 140 symbols. And it gets impossible to compute. So at the moment, the best we can do is say, yeah, we're going to rule those out because we can't compute it for the ones that we can, because these very, very long double occurrence words are a minority. They're less than 1% depending on the species, less than, point something percent. We're still missing because it could be that these very, very long double occurrence word would be outliers and they would pull all of my mean values up. But for the median, it would still be okay to compare.
Good. Thank you.
Nicole, your question?
Hi. Yeah, thanks for a really interesting talk. I was curious about the torsion that you found, and if you have an understanding of the structure that's giving rise to it, and just a little bit more about that.
So I can tell you that I found the generator with Sage, but it had so many vertices I couldn't, let's say, print it out, try to piece it together. It had somewhere like thousands of nodes.
Okay. Really large. Okay, cool.
Yeah, really large. So it would be great if we could find something smaller. I can construct them to be smaller, but they don't arise from double occurrence words.
Okay, cool. Thank you.
I have another question. So you have a post set. Another way that you can construct a complex from a post set is you can take the order complex, which is where the chains through the post set give you the simplices. I'm wondering how these compare, and in particular, if you run them on the post set you've been looking at. So if you take these tangled words and you compute the order complex of the tangled word, would you see kind of a similar high Betty numbers? Or I guess what I'm wondering is, what does this approach capture differently than the order complex?
I did not compare with the order complex. I compared it with p-path complexes. So that might be related, but not quite the same thing. And what they capture are different structures, right? So in the p-path complexes, at least all the cones, so pyramids with a square base, for example, would be considered cells. So those get filled in, but with the Prodsimplicial cells, that doesn't happen. And what we're really trying to capture with the Prodsimplicial cells, so the groundwork, how do I scroll faster? Is if I go way back to the assembly graphs, I just want to show you the two square paths that arise. So if you look at this reduction, this reduction by 1, 2, and 3, so the green vertices, can somehow happen independent of this one. And what I want to capture when I identify these two paths and I fill this in is like, it doesn't matter if I do this one first or that one first. They both land here.
Yes.
So we did want the squares.
Yeah.
Whether or not it makes the same kind of sense, especially for H2, it gets harder to identify what these pathways would mean. So for loops, I can say, yeah, they're definitely different paths to go from point A to point B. And they're not equivalent paths. But when I have non-trivial Betty 2 numbers, Betty 3 numbers, it's harder to interpret those. Possibly the order would give me more insight and I could compare and add it to be more of the topological signature of the word graph or of this like scrambling of the contigs.
Nice. Yeah, that makes sense. Cool. I have another question. No, that's one. So have you looked at, you kind of looked at these tangled words, they give really big Betty numbers. Have you looked at any way of kind of, is there any nice characterization of words that give, that are contractible, where the complexes are contractible? And this maybe relates to that question you said about how finding kind of moves that are homologically equivalent, because that's kind of like contractible things that you could contract with. Yeah.
So right now, I only have the trivial examples off the top of my head. I have not been able to find a good, let's say, theoretical way to say this kind of reduction always reduces to, always contracts the complex while preserving the homology. I would have hoped that that would happen, let's say, with the tangled chords, and it didn't happen. So now I'm worried that it also behaves in unpredictable ways with longer words. It doesn't quite follow this like subword behavior.
Mm-hmm. Cool. Nice. Thank you. Any other questions? No. What we'll do is we will stop the recording and then people can have an opportunity to ask any further questions they want as well. But I'll stop the recording now.